Welcome to this animated educational journey into the anatomy and physiology of muscular tissue. Muscle tissue is a richly vascularized, highly cellular tissue composed of elongated cells known as muscle fibers or myocytes. Inside these myocytes are specialized contractile proteins called actin and myosin filaments. By utilizing energy from ATP, these filaments slide past one another to generate powerful contractions and smooth relaxations. Beyond simply enabling body movements, muscular tissue serves essential functions. It stores and moves substances throughout the body, maintains upright posture, and generates vital body heat. The two fundamental characteristics of all muscle tissue are contractility, the ability to shorten forcefully, and conductivity, the ability to transmit electrical impulses. Based on anatomical location, structure, and functional properties, muscular tissue is classified into three distinct types: cardiac muscle, smooth muscle, and skeletal muscle. Let us explore each of these in detail. We begin with cardiac muscle, also known as striated involuntary tissue. As the name suggests, cardiac muscle is entirely unique in its distribution, found exclusively within the myocardium, or the muscular walls of the heart. Like skeletal muscle, it features prominent cross striations under the microscope, but functionally, it operates entirely involuntarily under the control of the autonomic nervous system. Structurally, cardiac muscle fibers are the smallest among the three muscle types. They are uninucleate, containing a single, centrally located nucleus, and they form a complex branching network. A defining and exclusive feature of cardiac tissue is the presence of intercalated discs. These are specialized transverse thickenings of the plasma membrane that unite adjacent branching fibers. Intercalated discs contain desmosomes, which provide structural strength during vigorous beating, and gap junctions, which act as electrical conduits. When a wave of contraction is stimulated in one fiber, gap junctions allow it to instantly spread to neighboring cells, enabling the entire heart to contract as a synchronized, powerful sheet of muscle. Next, we examine smooth muscle tissue, often referred to as visceral muscle tissue. This type is widely distributed throughout the walls of hollow visceral organs, including the lungs, stomach, intestines, urinary bladder, gallbladder, blood vessels, airways, eyes, and skin. Unlike cardiac and skeletal muscles, smooth muscle fibers completely lack cross striations, giving them a smooth, uniform appearance under a microscope. Each individual fiber is small, ranging from 20 micrometers to half a millimeter in length, and is spindle-shaped, or fusiform, featuring a thick middle portion that tapers into narrow ends. They are uninucleate, with a single prominent nucleus situated right in the center of the cell. In certain organs, such as the intestinal walls, smooth muscle fibers are extensively interconnected by gap junctions, allowing for powerful, coordinated contractions. Smooth muscle operates involuntarily, producing slow, sustained contractions that are highly resistant to fatigue. This specialized action is essential for regulating the lumen diameter of blood vessels, propelling food through the gastrointestinal tract via peristalsis, and expelling waste from the urinary bladder. Finally, we look at skeletal muscle tissue. This is the most abundant muscle tissue in the human body, constituting approximately 40% of total body weight and accounting for 1/3 of all body proteins. Skeletal muscles form the primary flesh of the body and contribute heavily to overall resting metabolism. They are directly attached to the bones of the skeleton through densely packed, white fibrous, non-elastic connective tissue structures called tendons. Structurally, skeletal muscle fibers are long, cylindrical, and arranged perfectly parallel to one another. They are the widest and longest fibers of the three types, with lengths ranging from a few centimeters to over 40 centimeters. Under the microscope, they exhibit beautiful alternating dark and light longitudinal and cross striations, which is why they are also called striped muscles. Unlike the other two types, skeletal muscle cells are multinucleated, with numerous nuclei located right at the periphery of the fiber, beneath the sarcolemma. Functionally, skeletal muscle is strictly voluntary, controlled directly by neurons of the somatic nervous system to facilitate locomotion, skeletal movement, and posture maintenance. To fully master the histology of muscular tissue, let us summarize the essential differences across the three types. First, regarding location, cardiac muscle is exclusive to the heart wall. Smooth muscle is located in visceral hollow organs and blood vessels, and skeletal muscle is attached to the skeleton. Second, looking at cellular appearance, cardiac fibers are branched and striated, smooth fibers are spindle-shaped and unstriated, while skeletal fibers are parallel, cylindrical, and heavily striated. Third, examining the nuclei. Both cardiac and smooth muscles are uninucleate with a central nucleus, whereas skeletal muscle is multinucleated with peripheral nuclei. Fourth, regarding specialized structures, intercalated discs are a unique hallmark of cardiac tissue, completely absent in smooth and skeletal muscles. And finally, in terms of neural control, cardiac and smooth muscles operate involuntarily through the autonomic nervous system, whereas skeletal muscle is under voluntary control via the somatic nervous system. Understanding these distinct structural adaptations clarifies exactly how each muscle type accomplishes its critical physiological role in the human body. Thank you for watching.